Welcome to a walkthrough of WEGAN's clustering and classification tools for analyzing ecology data. WEGAN is a web-based platform for analyzing ecology data that requires no coding knowledge. This step-by-step tutorial will show users how to use clustering and classification tools such as k-means, dendrograms, and heat maps. Visit wegan.ca to begin.
First, click the Clustering and Classification module to explore the groupings of communities and predict ecological community structures. Next, upload your dataset using the Choose File button. Alternatively, users can use sample datasets. For this example, we will select the dune sample dataset on grass species in a meadow. Selecting the dune dataset will also load the accompanying environmental data. Click the Submit button to process your data file. The Data Integrity Check page will confirm whether the data is usable, such as checking for correct sample and variable labeling. See our data uploading tutorial for more details on how to upload data and manage errors. Here, we see the dataset has passed the integrity check. Scrolling down, the Data Overview page will also display a PCA plot and heat map of your data. The Data Editor tab can be used to make slight modifications to the abundance data. Here we can change the data type, edit the data, and remove entries. Find more details on our data uploading tutorial. Once ready to continue, scroll down and click Proceed. On the normalization page, you can select the normalization methods appropriate for your dataset.
There are 4 manual data transformation options to choose from. Click a radio button to select a methodology. Click Manual Normalization to apply your own data normalization configurations. A pop-up will show you variable data distribution plots comparing the effect of your selections. In this example, no normalization was applied. To make revisions to the normalization, click Go Back. Going back, we can alternatively choose automatic normalization to apply recommended normalization settings based on Pearson mode skewness. For example, here are the distribution results of the automatic normalization.
Click Export to save this plot. Select the export format from a drop-down menu. We can select from PNG, TIFF, PDF, SVG, and PostScript files. Next, select your desired export resolution. Options range from 72 dpi to 600 dpi. Finally, select an export size of either full page or half page. Click Submit to generate the export file. The download link will now update. Click it to open the export file in a new tab. To move on from the normalization step, click Proceed.
For ease of replication, this tutorial will continue with no normalizations.
The second Data Integrity Check page verifies data quality in the supplementary data. This page is for our environmental data. Here, we see our supplementary data has passed the integrity check. Click Proceed to continue. After completing data pre-processing, you can select the type of clustering and classification analysis to perform. This example will start with the dendrogram plot. We have now reached the main analysis page. Here we can choose our desired plot and customize plot parameters. Start by adjusting the variables and parameters of the plot. First, select the data type from either normalized or original non-normalized data. Following that, select the methodology to use for measuring distance. Next, select which algorithm to use for clustering your samples. Finally, select a grouping variable for labeling samples. Click Update to regenerate the plot with your selected parameters. Click on Customize Graph to modify the appearance of the plot. Select from 3 color palette options. The plotting options allow you to rotate the dendrogram and to add sample names. Click Update to apply your customizations. Scroll down to view your customized dendrogram. Nodes that are linked sooner in the plot are more closely related or share more characteristics. Click Export to download your plot. Each unique plot should be saved using the Export button. After adjusting the export parameters, click Submit to update the download link. Continue to the k-means submodule from the navigation tree on the left. K-means is a clustering technique that separates data into distinct groups based on feature similarity. Each cluster is represented by a centroid, and data points are assigned to the nearest centroid. The centroid is then iteratively adjusted to reduce variance. Enter an integer value specifying the number of clusters to group. Click Update to apply your changes. Scroll down to view your k-means plot. There is one plot per cluster.
The x-axis shows all the species or features, and the y-axis provides intensity values. Each feature is related to each cluster at differing intensities.
Click Export to download your cluster analysis results. Another example shows
The feature data
in 6 clusters. Note that spacing restrictions will make large number of clusters difficult to visualize. Click the Results banner to expand a list of data files related to this submodule. Here we can view the detailed k-means data table.
A pop-up table shows us which features are categorized to which clusters.
Continue to the heat map submodule.
From the left-hand menu,
A heat map visualizes
data abundance patterns.
across samples and features. For this heat map, start by selecting the data type. Then select your preferred methodology for measuring distance. Select from 4 clustering algorithm options. Next, select whether to standardize your data through auto-scaling of features or of samples. Select whether to reorganize the feature or sample data if needed. Select whether to group the data and by which variable. Finally, select a test to perform and input an integer of how many significant features to show. Note that these
Selections only apply if
a grouping variable is selected. Click Update to apply your changes. Click to customize your graph. For the heat map, we can select what contrasting colors to use.
for the abundance gradient.
We can also adjust the overall palette. Other options include adjusting the borders showing the averages and labels. Click Update to apply your customizations. Scroll down to view your customized heat map.
Each colored cell on the map corresponds to an abundance value with samples in rows and features in columns. The heat map helps identify samples or features that are unusually high or low in abundance.
Remember to export your customized plot. Continue to the SOM submodule from the navigation tree on the left. A self-organizing map, SOM, is a neural network used for clustering and visualizing high-dimensional data. Data points are arranged on a grid and clustered based on similarity. Neighboring clusters have similar characteristics, creating a smooth transition across the map. Input an integer for clusters along the x dimension. Total clusters are calculated by number of x dimensions by number of y dimensions. Select a method for initialization. Input an integer for clusters along the y dimension. Total clusters are calculated by number of x dimensions by number of y dimensions. Select a neighborhood option. Bubble function is constant while Gaussian is decreasing. Click Update to apply your changes. Scroll down to view your updated SOM plot. This plot shows 2 x dimensions by 3 y dimensions for 6 total clusters. Click Export to save your plot. Under the Results banner, you can view the SOM data in table format. Click to open the table. The table highlights the SOM clustering details, showing which sites are assigned to which clusters. Continue to the PLSDA submodule to perform partial least squares discriminant analysis on your data. The partial least squares discriminant analysis, PLSDA, maximizes the covariance between the data matrix, X, and the group labels, Y. First, select a grouping variable. Next, select whether or not to keep class orders. If there are more than 2 group labels and the data do not represent quantitative differences, e.g., time series, disease severity, or treatment dosages, users should select Do Not Keep Class Orders. After, select the number of top components to display in the pairwise plot results. Click Update to apply your changes. Scroll down to view the PLSDA plot. Export to save your plot. Continue to the 2D Scores Plot tab from the top of the page. A 2D Scores plot visualizes how different samples, observations, are separated based on their latent variables, LVs, which are the linear combinations of the original features optimized for class discrimination. Select your LV1 x-axis component. Then select your LV2 y-axis component. Select whether to display the 95% confidence region. Select whether to display sample names. Finally, users can change the palette to grayscale. Click Update to regenerate the PLSDA scores plot. Scroll down to view the updated plot. Each point delineates an individual sample, with samples that are closer together being more similar. Different classes are color-coded to highlight class separation. The 95% confidence ellipses indicate the natural variability within each class. Good separation between groups suggests efficient differentiation between classes, while overlap between groups indicates high within-group variability or feature similarity. Click Export to download the plot. Under the Results banner, you can download the data file related to this analysis. Continue to the Loadings Plot tab. We again select our LV1 x-axis component and our LV2 y-axis component. Select whether to display variable names in the static plot download. Click Update. Scroll down to view the interactive Loadings plot. Loading plots interpret contributions of variables to latent components. Click on a point to view its details, highlight an area of the plot to zoom in, and double-click to reset the plot.
For example, clicking on the point at -0.16 and 0.53 opens a detailed box plot describing the data point. In another example, zooming in here
gives us this expanded view of the plot. Remember to export your plot. Under the Results banner, we can see a data file for download and can view a more detailed table of the plotted data. Click to view the detailed data table. Here we can see a detailed description of the sites and their position data. Click the Here link to download the table.
Additionally, clicking on a species name opens up a graphical summary, with original values on the left and normalized values summarized on the right.
Returning to the PLSDA page, we will now switch to the Cross-Validation tab. Cross-Validation assesses model performance. Select the maximum number of components to search. Next, select a cross-validation method, either tenfold cross-validation or leave one out cross-validation. Select a performance measure. Click Update to apply your changes. Scroll down to view your plot. Note that this plot is using the grouping variable selected in the Overview tab. Under the Results banner, you can view the detailed data table of the analysis results. This data table shows you more detailed statistics, including the accuracy,
R-squared and Q-squared measures for all plot components.
To elaborate on the grouping details, we can switch to the Overview tab. Here we will change the grouping variable from moisture to use. Click Update to re-execute the analysis using the new grouping variable. When we return to the Cross-Validation tab, our plot has updated. In another example, we will change the grouping variable further from use to moisture. This time, click Update on the Cross-Validation page. We immediately see our Cross-Validation plot update. Moving on, we will proceed to the Important Features tab. The 2PLSDA measures our variable importance in projection, VIP, and weighted sum of absolute regression coefficient, COEF. Start by selecting which measure to apply. If choosing VIP, select which component to score. If opting for coefficient, select the coefficient score. Input how many of the top variables to display in the plot. Finally, select whether or not to use grayscale. Click Update to apply your changes. Scroll down to view the Important Features plot.
The colored boxes on the right indicate the relative abundances of the corresponding variable in each group under study.
Click Export to download your plot. Under the Results banner, you can save the related data file or view a detailed data table of the plot. Click to view the data table. Here we see the detailed component 1 and component 2 values for each variable. Download this table by clicking the Here link.
Click on a feature name to view a graphical summary.
Returning to the main analysis page, continue to the Permutation tab. Permutation tests assess statistical significance by comparing observed statistics to a null distribution. A large sample size is required for reliable estimates.
Please note that a large sample size set with many permutations may take longer to process.
Select a test statistic using the radio buttons. Then set a number of permutations. Update to apply your changes. Scroll down to view the plot. Remember to export the plot. Continue to the PLSDA submodule. Sparse partial least squares discriminant analysis is used to reduce high-dimensional data for models that are easier to interpret. Start by selecting a grouping variable. Select the number of components. You can select to keep the same number of variables per component, or you can specify the number of variables for each component. Update to apply your changes. Scroll down to view the grid of plots. Click Export to download your plot. Proceed to the 2D Scores Plot tab. The 2D Scores plot visualizes data in 2 dimensions. Start by selecting the X component and the Y component. Select whether or not to display the 95% confidence region. Select whether or not to display sample names. And finally, select whether or not to use grayscale. Click Update to apply your changes. Scroll down to view the plot. Remember to export your plot. Find the downloadable data file related to this plot under the Results banner. Proceed to the Loadings Plot tab. The Loadings plot displays features important for group discrimination. Select which component to show loadings for. Click Update. Scroll down to view the loadings plot. The model selected variables for a given component are ranked by absolute values of their loadings. The selected component is along the x-axis, and the model selected variables are along the y-axis. The color legend shows the high to low color associations. Export to save your results. Under the Results banner, you can download the loading data file and view a detailed data table. Moving on, switch over to the Performance tab. To assess the performance, cross-validation is performed. Select from either the 5-fold cross-validation or the Leave One Out cross-validation. Click Update. View the classification error rates plot below. Click to export. Finally, we will continue to the OPLSDA submodule. To perform OPLSDA analysis, our dataset must contain a 2-group variable. We will therefore continue with the BCI dataset for this submodule with no normalizations applied. The orthogonal partial least squares discriminant analysis is a variant of the PLSDA technique for identifying patterns in data and predicting group memberships. This supervised classification method separates the predictive and orthogonal components for use in modeling the between-group and within-group differences, respectively. This model requires a binary response variable and is built to maximize the separation between the 2 groups. Start by selecting a grouping variable. You can also select plotting options. Click Update. Scroll down to view the 2D scores plot. The data points are either labeled as belonging, yes, or not belonging, no, to the grouping variable. Export to download the plot. Download the data file related to this analysis under the Results banner. Continue to the S-Plot tab. The Significant Features plot visualizes the variable influence in the OPLSDA model by combining covariance and correlation loading profiles. Select which labels to annotate. Click Update. Scroll down to view the S-Plot. Click on a point to expand its information. Expanded information for a specific point will appear in a second plot. Under the Results banner, you can download the data file related to this plot or expand a detailed data table of the plot results. On the Detailed Data Table page, click the Here link to download the table. Returning to the Analysis page, proceed to the Model Overview tab. The Model Overview tab provides a bar plot showing the
R-squared and Q-squared values for the model. A robust and predictive model should have high values in both R-squared and Q-squared, but a high R-squared with a low Q-squared may indicate overfitting.
Download the data file for this plot from under the Results banner. Finally, move on to the Permutation tab. Permutation tests assess statistical significance by comparing observed statistics to a null distribution. A large sample size is required for reliable estimates. Select the number of permutations from 100, 1000, and 2000. Click Update to apply your changes. Scroll down to view the plot. Click Export to download the plot. Access all data files from this analysis session through the Download option in the left-hand menu. Click Results to expand the list of files for download. A list of all data files from this analysis session will open. Click on any file to download. Because we restarted our analysis with the BCI dataset for the last submodule, we only see the OPLSDA files. The download.zip file contains all listed files. Download the WEGAN Docker file to recreate the WEGAN coding environment. Download the RSource file to get the exact R script used during your analysis session. After exporting your results, click Exit to return to the home page. We hope you found this tutorial on the clustering and classification module helpful.